Imagine knowing the exact identity of the assassin that is just slowly destroying your cells. Like you can map its structure out, you can see it operating under a microscope, and you know exactly how it's driving a disease.
Right.
But then when you try to send a drug in to stop it, you realize you literally can't touch it. It's completely smooth. There's, nowhere for a standard drug to grab onto.
Yeah, that is the classic biochemical nightmare of what researchers call undruggable proteins.
Exactly. And welcome to the Deep Dive. If you're joining us today, you are exactly like us. You're a curious learner trying to decode the invisible machinery that keeps us alive and, well, sometimes makes us sick.
Absolutely. And, it's one of the most frustrating barriers in modern medicine because for decades, drug design has really relied on a lock and key mechanism.
Right, where you find a little pocket, right?
Exactly. You find a crevice or a pocket on a disease-causing protein, and you engineer a tiny chemical molecule to fit perfectly into that pocket and just, jam the machinery.
But when you're dealing with proteins that lack those convenient little pockets, which, that happens constantly with cancers and neurodegenerative diseases, that entire model just falls apart.
It does. It completely falls apart.
Which brings us to our mission today. We're exploring a radically different approach hidden in our latest stack of research. We are looking at a completely new way to hunt down and destroy these smooth, undruggable proteins right inside the fluid of our own cells.
And crucially, without having to genetically modify the patient at all, we are talking about a massive paradigm shift here called targeted protein degradation, or TPD.
TPD, right.
Yeah, the philosophy shifts from block the protein to destroy the protein. Instead of trying to jam the gears, TPD aims to hijack the cell's natural garbage disposal system and physically drag the bad protein into the shredder.
Which sounds incredibly elegant in theory. And we actually already have the perfect tracking devices for these bad proteins, right? Antibodies. They're the ultimate biological bloodhounds.
Oh, they're amazing. You can program them to latch onto almost anything with just absolute precision.
But there's a catch. Why haven't we just been injecting antibodies into our cells to clear out these bad proteins all along?
Well, it comes down to the physical realities of the cell. Antibodies are massive, bulky, Y-shaped molecules. They're fantastic at floating around in your bloodstream, tagging viruses on the outside of cells.
Right.
But getting them through the fatty, tightly guarded outer membrane of a cell and into the interior cellular fluid, the cytosol, that is incredibly difficult.
Because if you try to force them in, they usually denature, right? They lose their shape and just become totally useless.
Exactly. They get crushed by the environment.
Okay, let's unpack this. We have this powerful weapon, but it's too big to fit through the door, and the environment inside degrades it. So the research we're exploring today proposes this fascinating workaround.
Right. Instead of altering the cell or altering the antibody, the scientists built a microscopic liquid delivery vehicle.
Yeah, they found a way to wrap the bulky antibody inside a synthetic fluid droplet that basically escorts it safely into the cell.
It's brilliant. They rely on a biological phenomenon known as liquid phase separation, or LLPS. And the end result is something they call a biomolecular condensate, or a BMC.
I want to pause on this concept of phase separation because it's so crucial to really getting this. Think about making a balsamic vinaigrette in your kitchen.
Right, the oil and the vinegar.
Exactly. You pour them in, and they naturally separate into distinct droplets, right? The oil beads up inside the vinegar. In this case, the scientists are mixing highly specific molecular ingredients that spontaneously bead up, trapping the antibody inside the quote-unquote oil drop.
Yeah, but it's a highly engineered vinaigrette. The mechanism here comes down to designing specific chains of amino acids, which we call peptides, with opposing electrical charges.
Okay, so a positive and a negative.
Exactly. They created two distinct peptide chains. One is an arginine-rich precursor, which they write out as C, little CR8, C, then a GG spacer followed by RR or G. And that one carries a very strong positive charge from its guanidinium groups.
Wow, okay. And the other one?
The other is made of 10 aspartic acids, called D10, which carries a strong negative charge from its carboxylate groups.
Wait, hold on. The inside of a biological system is essentially a salty soup, right? It's full of freely floating ions and competing charges. Wouldn't these positive and negative peptides just get neutralized or disrupted by, everything else floating around?
You'd think so, but that's the brilliance of the engineering. They aren't just relying on a mild static cling here. When those specific guanidinium and carboxylate groups meet, they form incredibly dense electrostatic networks.
So it's a really tight grip.
Incredibly tight. The binding force between them is remarkably strong. In biochemical terms, they have a dissociation constant, or KD, of 4.3 micromolar. They pull together with a tightness that literally forces the surrounding water molecules out of the way.
So it's less like a loose drop of oil and more like a molecular flash mob. These positively and negatively charged peptides are programmed to instantly rush together, lock arms, and form this tightly packed sphere.
Yeah, exactly.
And because they're doing this in the presence of the antibodies just get caught right in the middle of the huddle.
Precisely. And because that electrostatic grip is so strong, this flash mob can hold an enormous amount of cargo. The data actually shows they can encapsulate 289.4 micrograms of the antibody per 100 micrograms of the polyarginine peptide.
Oh, wow. So they're packing in nearly 3 times their own weight in antibodies. That is a remarkably efficient microscopic delivery truck.
It really is.
Okay, so to quickly recap for you, our listener, before we move to the next layer of this technology.
Yeah.
Bullet 1. TPD aims to clear bad proteins inside cells, but delivering bulky antibodies is really hard.
Right.
Bullet 2.
Yeah.
Scientists are using liquid phase separation to trap antibodies in specialized droplets called biomolecular condensates. And bullet 3. This process is driven by positive arginine and negative aspartic acid peptides locking together. So, question for you to think about. If you were designing a microscopic delivery truck, how would you keep it from melting into the rest of the cellular fluid before it reaches its destination?
And that question brings up a massive problem, right? If we think about the physics of liquid, a raw droplet of liquid is inherently unstable.
Right. If you have a bunch of these liquid flash mobs floating around and they bump into each other, wouldn't they just merge? Like, I imagine they coalesce into one giant useless puddle and just drop all the antibodies before they ever reach the target cell.
You've hit on the exact reason why raw biomolecular condensates usually fail in medical applications. Coalescence is fatal to the delivery process. So to solve this, the researchers had to give the liquid droplet a suit of armor.
Armor. I love this.
Yeah, they coated the raw droplets in a metal-phenolic network, or an MPN shell.
Okay, let's unpack how this armor actually works. What exactly is a metal-phenolic network?
It's a chemical mesh made from 2 very simple ingredients. The first is EGACG, which is actually an organic plant-based compound. It's the main active antioxidant you'd find in green tea.
Oh, wow, green tea. Really?
Yeah. And the second ingredient is iron, specifically iron chloride ions. When you introduce these 2 elements to the outside of our liquid droplet, the iron ions act like microscopic staples, physically linking the green tea molecules together into a tight, semi-permeable cage around the liquid.
Okay, so think of it like an M&M candy. The biomolecular condensate is the soft chocolate center, and the MPN is the hard candy shell that stops it from melting in your hands.
That is a perfect analogy.
And the data shows this shell does something incredible. It physically squeezes the droplet down. The raw liquid droplets are quite clunky, right? Measuring about 2 micrometers wide.
Right.
But once that iron mesh forms around them, it condenses them into sleek, nanoscale spheres roughly 300 nanometers wide.
Yeah.
Why does shrinking them to that specific size matter so much?
Because size dictates how a cell interacts with foreign objects. A 2-micrometer droplet is just too big. When it hits a cell, it tries to passively fuse with the cell membrane, which is a slow, inefficient process that can actually damage the cell.
But 300 nanometers.
That hits the exact physiological sweet spot for a process called active endocytosis.
Meaning the cell actively swallows it?
Exactly. The cell membrane naturally folds inward, wraps around the 300 nanometer droplet, and pulls it inside in a little transport bubble called an endosome, and the efficiency difference is staggering.
Let's hear the numbers.
The data reveals that 99% of these armored nanodroplets are successfully swallowed by the cells within just 4 hours.
99%. Just for context, the commercial delivery agents on the market right now, like XFect, they hit maybe 42%. And Project is at 32%. So 99% is almost unheard of.
It's a massive leap forward.
But I have to ask, if the cell swallows this armored M&M in a transport bubble, how does the antibody break out of the armor and actually do its job?
That's where the chemistry of the iron phenolic armor gets really elegant. The armor is highly pH responsive. When a cell swallows something into an endosome, it doesn't just leave it there.
Right, it wants to digest it.
Exactly. The cell starts pumping protons, which is basically acid, into that bubble to digest whatever it just ate. As the endosome matures, it turns into a lysosome, and the pH drops to a highly acidic 5.4.
And what does acid do to the iron staples holding the armor together?
It breaks them. The influx of protons fundamentally disrupts the coordinate bonds between the iron and the organic molecules. Essentially, the acid rusts the staples out of the corset. The mesh falls apart, the liquid droplet dissolves, and the antibody is unleashed right into the cytosol.
Wow, and the data backs this up, right?
Oh, yeah. They found that at that lysosomal pH of 5.4, 80% of the cargo releases within 15 hours, but at a neutral pH, it releases much, much slower.
That is fantastic. So, to synthesize the journey so far, we've trapped a precise antibody in a liquid droplet, armored it in an iron mesh that shrinks it to the perfect size to trick the cell into swallowing it, and engineered the armor to dissolve only when it hits the acid of the cell's stomach.
Exactly.
But once it's loose inside the cellular fluid, we still have a problem. The antibody knows how to grab the bad protein, but how does it know where the cell's trash compactor is?
It requires a navigation system, and the researchers built that right into the original positively charged peptide we discussed earlier.
The arginine one.
Right. Along with the structural components and a cell-penetrating domain called cyclic octarginine, they added a specific sequence of 4 amino acids to the tail end. Arginine, arginine glycine, or RRRG.
Separated by that GG flexible spacer we mentioned.
Yeah.
What does the RRRG do?
It acts as a built-in GPS tag. Biologists call it a proteasome-targeting motif, or PTM. We'll get into exactly how it plugs into the cellular machinery in a moment, but essentially, this 4-letter chemical sequence is the address label that routes the entire payload straight to the cellular shredder.
Okay, so time for another quick 5-minute recap for you. Bullet 1. MPN shells made of EGCG and iron act as armor to stabilize the droplets.
Yep.
Bullet 2. The shell shrinks them to the nanoscale, boosting cell uptake to a whopping 99% via active endocytosis.
Right.
And bullet 3. An RRG sequence acts as a GPS to route the payload to the cell's trash compactor. So here is your next question to ponder. Now that our armored GPS-guided droplet is inside the cell, how does it know to destroy the disease without hurting healthy tissue?
And that is the multi-million-dollar question.
Here's where it gets really interesting. Let's look at how it performs in the real world. The source material highlights an ultimate test case targeting a notorious mutant protein called KRAS. For anyone unfamiliar, why is KRAS the ultimate boss fight for this technology?
KRAS is a 21 kilodalton cytosolic signaling protein that acts like a cellular light switch for growth. When it mutates, the switch gets jammed in the on-end position, leading to rapid, uncontrolled cell division, which, as we know, is cancer.
Right.
And it is the classic undruggable protein because it has that perfectly smooth surface we talked about. But the specific mutation they targeted, KRAS G12V, makes this experiment incredibly dangerous.
Dangerous.
Because the G12V mutation means there is only a single amino acid difference between the deadly cancer-causing protein and the completely healthy normal KRAS protein that your non-cancerous cells desperately need to survive.
Oh, wow.
Yeah. If your delivery system isn't perfectly precise-like, if it accidentally degrades the healthy wild-type KRAS alongside the mutant KRAS, you will kill healthy tissue and potentially kill the patient.
So the precision has to be absolute. And the researchers proved this by setting up this fascinating high-stakes co-culture experiment.
They did.
They took 2 completely different populations of human cells. One group, called SW480 cells, carried the deadly mutant G12V KRAS. The other group, HT29 cells, carried the healthy, normal KRAS. They mixed them together in the exact same dish, dropped in our armored droplets loaded with a variant-specific antibody, and just watch what happened.
And the selective targeting was flawless. The droplets completely ignored the healthy cells. But in the mutant cancer cells, they seized the G12V protein and rapidly degraded it. Normally, this cancer protein survives in the cell for about 22 to 24 hours.
Okay.
The treatment slashed its half-life to less than 12 hours. It was literally being cleared out faster than the cancer could produce it.
But, a petri dish is a very controlled environment. When you take a complex system like this, iron-coated droplets carrying giant biological molecules and inject it into a living, breathing body, things usually go wrong. Did they test this in a live model?
They did. They used a mouse xenograft model, meaning they grew human KRAS-driven tumors inside mice and then injected these condensates directly into the tumors at a dose of 10 milligrams per kilogram 3 times a week.
And we aren't just looking for a slight reduction in tumor volume here, right? We need to know how the tumor is reacting.
Exactly, and the pathology reports were definitive. They didn't just measure the size, they measured the specific birth and death rates of the cancer cells. They looked at apoptosis markers using caspase-3 and tunal staining, which basically measures programmed cell death, and those were massively elevated.
So the cells were actively dying.
Yes, and at the same time, markers for cellular proliferation, specifically Ki-67, meaning how fast the cancer is dividing, those plummeted. The tumors weren't just pausing, they were actively dying from the inside out.
Which brings up my biggest concern, toxicity. We're injecting iron and foreign peptides into a mammal. When they analyzed the mice, did they find collateral damage in the liver or the kidneys?
That's maybe the most remarkable part of the safety profile. There was zero systemic toxicity. The mice maintained normal body weight. And when they ran blood panels checking 15 different critical serum biomarkers for liver and kidney function enzymes and all that, every single one came back completely normal.
That's wild.
Yeah.
And that loops right back to the pH-responsive armor we discussed earlier. Because the iron shell only breaks apart in the highly acidic environment of a cellular endosome, the droplets stay locked up tight while they are in the neutral pH of the bloodstream. They just don't leak.
Exactly. It's a highly contained reaction.
Okay, let's do our third recap here. Bullet 1. The antibody-loaded condensates can differentiate between a healthy protein and a single mutation cancer protein.
Yep.
Bullet 2.
Yeah.
In cell models, it rapidly accelerated degradation of the cancer-causing KRAS G12V.
Right.
And bullet 3. In mouse models, it effectively suppressed tumor growth without systemic toxicity. So here's your next question. We know the cancer protein is disappearing, but what exactly is the cell using to shred it to pieces?
Right, because to truly understand why this platform is a paradigm shift, we need to look closer at what happens right at the end of the line.
Yeah, how is the actual killing happening? We know the GPS tag routes the protein to the trash compactor. But cells actually have 2 different recycling systems, right? Autophagy, which is like tossing bulk waste into a vat of acid, and the proteasome, which is a highly regulated mechanical shredding machine.
Exactly, and it's vital to know which one is doing the work. To figure this out, the researchers used molecular blockers. They drugged the cells with chloroquine to shut down the autophagy vat, and the cancer protein still got destroyed.
Okay, so it's not autophagy.
Right, but when they used a specific chemical called MG132 to jam the gears of the proteasome shredder, the destruction completely halted.
Ah, so this proved the degradation is entirely proteasome dependent.
Exactly. But how it connects to the proteasome, what's fascinating here is how groundbreaking this mechanism actually is. Normally, for a cell to send a bad protein to the proteasome, it relies on a complex biological tagging system called ubiquitination.
Let's explore that, because ubiquitination is usually a massive hurdle in these kinds of drugs. How does it normally work?
Well, in a standard cell, if a protein is damaged, specialized enzymes called ligases have to come along and physically attach a long chain of ubiquitin molecules to the bad protein. It's like sticking a dozen brightly colored flags onto a piece of trash.
Right, and the shredder won't open without the flags.
Exactly. The proteasome only opens its doors if it sees those ubiquitin flags. Engineering this flag-tagging process for a new drug is incredibly complex, and it often requires genetically modifying the cell's enzymes to recognize the target.
But our armored droplet doesn't do any of that, does it? It bypasses the flag system completely.
It completely bypasses the entire ubiquitin pathway. Remember that RRRG tag on our droplet?
The GPS.
The GPS. Through molecular docking analysis, the researchers found that this specific 4-amino acid sequence acts like a universal key that fits perfectly into a receptor on the outer lid of the proteasome, specifically called the PSMD9 subunit. Yeah, it binds with a powerful free energy of negative 7.7 kilocalories per mole. Just for comparison, they tested a dummy sequence, AAG, and that barely bound at all with a weak negative 4.6.
So it's a direct molecular bridge. The antibody side of the droplet grabs the cancer protein, and the RRRG side of the droplet physically plugs straight into the shredder's entry port, the PSMD9, forcing the protein inside. No genetic modification, No ubiquitin flags required.
Exactly. And because it's a universal bridge, the entire platform is modular. If you want to cure a completely different disease, you don't have to redesign the chemistry of the droplet or the GPS tag. You just swap out the antibody payload.
Which is huge.
It is. And to prove this modularity, the researchers targeted a completely different disease driver, alpha-synuclein.
Alpha-synuclein is a major player in neurodegenerative diseases, particularly Parkinson's. And it presents a totally different set of challenges than the KRAS cancer protein, right?
Oh, totally. For one, alpha-synuclein is a 17-kilodalton protein that tends to aggregate inside the nucleus of neurons, which is even harder to reach than the cellular fluid.
Right. Furthermore, in diseases like Parkinson's, the cell's natural autophagy systems are often impaired. The cell simply loses the ability to clear out the toxic buildup on its own. So they took an SKNSH human cell model, basically neuron-like cells, overloaded them with toxic alpha-synuclein, and treated them with droplets carrying an anti-alpha-synuclein antibody.
Did it cross into the nucleus?
It did. They literally used advanced imaging techniques that light up under a microscope, and they could visually see the droplets dragging the alpha-synuclein directly into the proteasome shredders right there in the nucleus.
And what was the impact on the dying neurons?
It profoundly reversed the neurotoxicity. In the untreated cells, the toxic buildup was killing them rapidly. Cell viability was down to a dismal 32%.
Oh, wow.
But after a single treatment with the targeted droplets, viability rebounded to nearly 79%. And they measured LDH release, which is a marker of cell damage, and that dropped by 28%, returning to normal levels.
That's amazing. Yeah, and they even used label-free quantitative proteomics to confirm that the clearance was happening specifically in the exact subcellular compartments needed. They essentially rescued the dying neurons by clearing the toxic debris.
That is just phenomenal.
Okay,
our final recap here for you.
Yeah.
Bullet 1. The system works independently of standard ubiquitin tagging, directly docking to the proteasome via PSMD9.
Yep.
Bullet 2. The modular design means you can just swap the antibody to cure a completely different disease.
Right.
And bullet 3. It successfully cleared alpha-synuclein, rescuing dying neurons and proving it works on nuclear targets too. So final question for you. If you could load these microscopic droplets with an antibody for any protein in the human body, what disease would you target first?
It really opens up endless possibilities.
It does. So what does this all mean? To step back and look at the whole picture we've painted today, we are looking at a system that takes off-the-shelf antibodies, wraps them in a stabilized nanometer scale metal-phenolic corset, safely delivers them right into the cell, and acts as a direct bridge to the cell's own destruction machinery.
Right. It's an all-aqueous, genetically unmodified modular delivery system.
Exactly. It's modular, it requires zero genetic modification, and it works across entirely different classes of disease.
If we connect this to the bigger picture, we have to recognize how much time and resources have been spent over the last few decades trying to design tiny chemical drugs to fit into the microscopic, hidden pockets of proteins.
Billions of dollars.
Billions. It's been incredibly difficult work, and for many diseases, it has repeatedly failed. But if biomolecular condensates can simply encapsulate our most powerful pre-existing antibodies, carry them through the cell wall, and force the target right into the shredder, well, the very concept of an undruggable target might be completely obsolete.
Imagine a near future where a doctor tells you that the protein causing your disease is undruggable. And what that actually means is we just haven't packed the right droplet for it yet.
Exactly. It completely rewrites the rules of engagement for intracellular disease.
It really does. Thank you for joining us on this deep dive into the microscopic front lines of biotechnology. Keep questioning the established rules, keep exploring the invisible machinery that makes us who we are, and we'll catch you next time.